Hello everyone, welcome back. As we discussed earlier, body cells have different capacity to regenerate following injury. The regenerative capacities of tissues or organs depends on their cellular composition. We now continue to talk about the regenerative process in different tissues and fibrous repair. After this class, students should be able to define granulation tissue and sketch its microscopic appearance. Although a covering epithelium has a strong capacity to regenerate, this depends upon the relationship with the adjacent connective tissue that provides mechanical support and factors regulating the epithelial growth and function. For example, if the skin epidermis is damaged, but the underlying basement membrane is intact, then the epidermal basal cells will proliferate and migrate into the wound, resulting in perfect restoration. And how about regenerative process in granular epithelium after injury? As shown in this diagram. If the underlying basement membrane is not involved, the residual granular cells will proliferate to establish the normal tissue structure and function. Normally, hepatocytes are quiescent cells with strong capacity to regenerate following injury. If the damage only affects parts of the parenchymal cells in the hepatic globule, as in acute hepatitis, the normal architecture and function will be restored through proliferation of healthy hepatocytes. By contrast, if the liver suffers extensive destruction with collapsed reticular network, repair could only be achieved by scar formation even if the remaining hepatocytes have good reparative potential. Fibrous tissue cells have a marked regenerative ability. This diagram shows the maturation process of fibroblasts. In response to injury, the mesenchymal stem cells can differentiate into active fibroblasts to synthesize new extracellular matrix and ground substance. Fibroblasts are ovoid or satellite cells with long, tapering processes. They have one elliptical nucleus with one or more clear nuclei. Active or immature fibroblasts usually have weakly basophilic, relatively visible cytoplasm. With time, fibroblasts gradually mature into fibrocytes, showing elongated nucleus with weakly acidophilic and scanty cytoplasm. At the same time, we can see abundant collagen deposition around the fibrocyte. Proliferation of fibrous tissue along with capillaries to form granulation tissue is critical to fibrous repair. We shall discuss the regenerative process of capillaries. Please note this process in capillaries differs from that in large blood vessels. Regeneration begins with the enzymatic dissolution of basement membrane in the existing capillaries, and followed by the proliferation of endothelial cells. This leads to the formation of solid endothelial cell sprouts, which then migrate the injured site, differentiate, and begin to form vascular channels. These small channels then join with nearby capillaries to form bigger vascular channels. Initially, the newly formed vessels have gaps between the endothelial cells and poorly formed basement membrane. To adapt to the functional needs, some blood vessels acquire a muscular coat to become arterioles, while others enlarge to form thin wall venules. After injury, the endothelial cells of larger blood vessels proliferate and have the ability to re-establish an intact inner lining. But smooth muscle cells in the vascular walls do not have significant regenerative ability. Therefore, the wounded smooth muscle layer is often replaced by scar tissue. Now, we have finished discussing the regenerative process in different tissues. Next, we'll proceed to fibrous repair. Tissues that cannot be replaced as good as new by regeneration must be patched up by fibrous repair or scar formation. Scarring occurs if the tissue injury involves the supporting stromal framework or if the injured tissue is composed of permanent cells. Fibrous repair starts with the formation of granulation tissue and culminates in the laying down of fibrous tissue. Basically, fibrous repair occurs in the following sequence. The first step is the formation of new blood vessels to nourish this process. And then the fibroblasts will proliferate and migrate into the injured site and synthesize collagen fibers and extracellular matrix to knit together the disrupted tissue. Subsequently, the fibrous tissue continues to be modified and remodeled. We have repeatedly mentioned the term granulation tissue, but what does it really mean? What are its morphology and functional roles in fibrous repair? Granulation tissue is a fragile, easy to bleed, highly vascular immature connective tissue, characterized by production of fibroblasts and newly formed thin wall capillaries in a loose extracellular matrix often mixed with inflammatory cells. On close inspection, granulation tissue is pink, soft, moist, and has a granular appearance. Its histologic appearance is characterized by a mixture of proliferating newly formed capillaries, fibroblasts, and inflammatory cells. These new capillary endothelial cells are swollen and its lumen is narrow. These capillaries are leaky because of incompletely formed interendothelial junctions. This leakiness explains why new granulation tissue is often edematous. These blood vessels will soon differentiate into muscular arterioles, thin wall venules, or true capillaries. The main function of fibroblasts is to synthesize extracellular matrix and growth factors. Granulation tissue have several properties to help in tissue repair. Firstly, granulation tissue formation can fill the tissue defect and protect the wound surface from infection and further injury. Secondly, the ingrowth of granulation tissue can replace or encapsulate the necrotic tissue, thrombus, inflammatory exudates, and foreign bodies. Here we emphasize the loss of granulation tissue because its formation is important to wound healing in most clinical situations. However, granulation tissue does not exist for a long time. Over time, the fibroblasts produce more collagen and then differentiate into fibrocytes. As maturation proceeds, fluid is gradually restored. Inflammatory cells disappear, and the site is occupied by less cellular, more collagenous tissue. Blood vessels shrink or disappear as the demand decreases. At this point, granulation tissue becomes scar tissue. These two pictures show the growth and histologic appearance of scar tissue. Macroscopically, scar tissue is tough and pale due to increased collagen and decreased vascularity. Histologically, scar tissue is mainly composed of fake collagen deposition. Unlike granulation tissue, there are usually no blood vessels or inflammatory cells. Sometimes, the scar form is excessive and ugly. Redundant accumulation of collagen in fibrous repair may result in keloid formation. Keloid refers to excessive collagen deposition in the skin forming a prominent raised scar, as seen in this picture. Some people have a genetic predisposition to keloid formation. Now, we have discussed the general principles of regeneration and fibrous repair. During epithelial regeneration, the residual epithelial cells from the margin proliferate and migrate into the wound if the underlying basement membrane is undamaged. Otherwise, fibrous repair would take place. Granulation tissue formation plays an important role in fibrous repair. Histologically, granulation tissue is composed of fibroblasts, newly formed capillaries, and inflammatory cells. As maturation and remodeling ensue, granulation tissue accumulates collagen and eventually turns into scar tissue. In the next section, we will talk about wound healing and factors influencing tissue repair. Here are the references, and see you next time.